Dear students, today we are going to deal with one of the non-thermal processing called ohmic heating. As we all know, heating is one of the ancient forms of food processing and preservation. It has been used by mankind for past many years. The ultimate aim of any conventional thermal processing of foods is to ensure microbial safety, enhance shelf life of food through destruction of enzymes, toxins, etc. Though the process is effective from microbial safety standpoint, they cause deterioration in sensorial and nutritional attributes of foods. Hence, to overcome the degradation of sensory characteristics such as flavor, aroma, texture and appearance, and also nutritional reduction, marvelous advancements with the development of novel thermal processing has been achieved. All the advanced methods of processing are highly energetic and efficient as the rate of heat transfer is very rapid. These are called novel thermal processing technologies. Among the novel thermal technologies like microwave heating, radio frequency heating, inductive heating, ohmic heating seems to be highly promised technique, especially in food industries. So, on completion of this module, you will be able to identify the principle behind ohmic heating in food processing and also understand the effect of ohmic heating on quality of foods. Let us first discuss about what is ohmic heating before going into its principle. Ohmic heating is defined as a process in which electric current is passed through materials with the purpose of heating them. Ohmic heating is also referred by other names such as electrical resistance heating, Joule heating, electroheating or electroconductive heating. Ohmic heating is different from other electrical heating methods. In earlier days, application of electric current in food processing was developed in pasteurizing milk and the process was called as electropure process. But later, the process was discouraged due to high processing cost. Again in 1980s, researchers began investing the potential of ohmic heating in food processing. We are all well aware about the conventional heating methods. But the thing is, ohmic heating is entirely different from the conventional heating. We will see the reasons behind this difference. Due to slow conduction and convection in conventional canning, significant damage to the product quality occurs, whereas in ohmic heating, the entire mass of the food gets heated due to volumetric heating. Thus, it resulted in better product quality. And also, it is possible to process large particulate foods which are difficult in conventional methods. Cleaning requirements are quite less than the conventional heat exchangers. So, now you got an idea on what ohmic heating is about, right? Let us now see the principle of ohmic heating. When an alternating electric current is passed through the conductive material of resistance, heat is generated internally due to electrical resistance causing temperature rise. Here, the food acts as an electrical resistor. So, from the figure, you can visualize the electrical circuit which represents the ohmic heating along with its electrical analog. Ohmic heating deals with electrical conductivity properties of foods. The ohmic heater contains electrodes which are in contact with food material, unlike in microwave and inductive heating where electrodes are absent. Also, the frequency applied is less compared to that of radio or microwave frequency range. Heat is generated within the food by the passage of an alternating electric current through a continuously flowing stream of food fluid. As heat is generated internally, ohmic heating offers the use of HTST technique, that is high temperature short time technique on particulate foods. The particles can be made to heat at the same rate or faster than the carrier liquid. Solids can be thus successfully treated in the short time required to maximize product quality. Ohmic heating possibly produces a sterile product which can further be moved to a sterile packaging, hence It is also referred as aseptic processing. Thus, it helps to preserve the perishable food material for a long duration of time. Electrical resistance heating allows particles and liquids to heat at the same rate and facilitates rapid heating of mixtures with high solid fractions. To understand the principle behind ohmic heating, it is necessary to study the electrical conductivity of food materials and predict the resulting heating patterns. Using ohmic heating, the foods can be pasteurized or sterilized or fermented similar to the current available methods of processing, which enhances the sensorial and microbial quality of foods in an appropriate manner. The ultimate aim of processing technique is to inactivate the microbes and enzymes. So, let us review the effect of ohmic heating on microbial inactivation. As we said already, ohmic heating is a high temperature, short time sterilization process. Microbial inactivation during ohmic heating is primarily due to thermal effects in nature. The principal reason for this added effect of ohmic treatment may be its low frequency, usually 50 to 60 Hz, which allows the cell wall to build up charges and form pores. The application of electric current tends to disturb the integrity of bacterial membrane and cause electrolysis of molecules on the cell surface. The mild electroporation during ohmic heating may contribute to the cell inactivation. Electroporation is the development of holes in the cell membrane due to individual ion pressure, which cause the change in permeability of the cell membrane due to the varying electric field. It is related to charge transport, which results from differences between the internal and external potential of cells when it is subjected to electric fields. If the electric field strength is exceeded the critical value, the membrane is permeabilized by pore formation. This permeabilization can be reversible or irreversible depending upon the electrical field strength, process time, cell size, membrane surface charge and suspending liquid medium. Corresponding electroporation also ruptures the structure of food sample during ohmic heating. It is sometimes considered desirable and sometimes unacceptable. Study shows that spore inactivation during ohmic heating was primarily due to the thermal effect, but there was an additional destruction caused by the electric current, which lead to spore injury and thus made them vulnerable to ohmic heating. The increase in lethality caused by the double heating is also known as the tyndallization effect. The enhanced spore inactivation by ohmic heating resulted in a greater rate of spore inactivation during the first stage of heating when compared with conventional methods. It also resulted in greater decrease in the count of viable spores immediately after the incubation period. For example, in milk sample, the inactivation of E. coli was reported to be faster in ohmic heated sample than the conventional heated sample. The application of electric field imparted a non-thermal additional killing effect over vegetative cells of E. coli in addition to the thermal effect. Similarly, we will see the effect of ohmic heating on enzyme inactivation. The presence of enzymes in food has either negative or positive impacts on food quality. In many food processing techniques, the control of enzyme activity is necessary to enhance or prevent enzymatic activity. A study on the impact of electric field on enzyme shows that the inactivation of polyphenol oxidase and lipoxygenase require shorter treatment time than conventional heating for the same degree of inactivation. Thus, the ohmic heating process reduces treatment time, which in turn reduces the negative thermal effects such as thermal destruction of vitamins, pigments and texture of food product. Similarly, ohmic heating treatment of fresh orange juice reduced the activity of pectin esterase to about 90 to 98%, whereas in conventional pasteurization process, the residual pectin esterase activity was reduced by 95% only. Likewise, the deactivation of pepsin activity was reported for grape juice as a function of electric field strength, electrical conductivity and pH. Whatever may be the processing techniques, either conventional or modern methods, it will have its own impact on the quality of food. The next topic is about the effect of ohmic heating on food properties. The degradation of anthocyanin was studied in both ohmic and conventional heating. Degradation increased with increasing voltage and solid content. At lower voltage levels, the degradation was lower than conventional heating and also exhibited higher anthocyanin degradation with higher electric fields. In a similar way to conventional heating, ohmic heating promoted degradation of ascorbic acid in orange juice. At 42 volt per centimeter of electric field strength, degradation of vitamin C was approximately 35% after 3 minutes of heating at 90 degrees centigrade. On comparison with other methods such as conventional, microwave and infrared heating, ohmic heating resulted in maximum retention of vitamin C. It was studied that ohmic heating caused significant changes in physical properties including viscosity, heat capacity, thermal and electrical conductivity of foods. It has been reported that conductivity decreased with degree of gelatinization, obviously caused by structural changes and increase in bound water. So far, we dealt with the principle and effect of ohmic heating process. Now, we will move on to the ohmic heating equipment. APV Baker ohmic heating process is a continuous type ohmic heater designed by United Kingdom Electricity Research and Development Centre in 1983. The equipment consists of feed pump, heating, holding and cooling section, series of electrodes and an aseptic packaging plant. Food is pumped through a vertical pipe containing a series of electrodes. Piston pump is preferred to allow high viscous food and particles to pass through with minimal damage. The electrodes are connected to 50 Hz 3-phase supply. Selection and design of electrodes must be carefully done to achieve higher degree of hygienic standards. Specially designed food compatible graphite electrodes are used to avoid metal dissolution by electrolysis. Electric current is then passed through the foot in the pipe connecting the electrodes. The food is rapidly heated to sterilization temperature in heating section and passed to the next holding section and then to the cooling section. Finally, the food material is passed to an aseptic packaging plant. The ohmic heating process provides a rapid sterility and is not lost due to the immediate packaging process. The system power is 10 kW, allowing a 75 degree centigrade temperature increase with a flow rate of 100 kg per hour. In ohmic heating process, particulates up to 25 mm are heated to sterilization temperature up to 140 degrees Celsius in less than 90 seconds, after which they are cooled back down to ambient temperatures within 15 minutes. These processing times are significantly shorter than the typical 2-hour process cycles for an in-can sterilization techniques. Commercially available ohmic heating equipment design place electrode at various positions either along the length of product flow, that is inline field, or perpendicular to the length of product flow or cross field, differing predominantly in distribution of electric field strength. it is must to know about the factors influencing efficiency of ohmic heating. The efficiency of ohmic heating depends on the rate of heat generation in the system, the electrical conductivity of food, electrical field strength, residence time, and the method by which the food flows through the system. Also, particle size, concentration, ionic concentration and electrodes have been found to influence the rate of ohmic heating of food. First and foremost factor is the electrical conductivity of foods. The food product contains the ionic species such as salts and acids which acts as electrolytes so that an electric current can be made pass through the food and generate heat inside it. In case of thermal processing, thermal conductivity of a product is the controlling factor, whereas in ohmic heating, it is the electrical conductivity of the food product. The design of an effective ohmic heater depends on the electrical conductivity of foods. The rate of ohmic heating is directly proportional to the square of the electrical field strength and electrical conductivity. The electrical conductivity of food tends to decrease with increase in temperature after the commencement of bubbling and with increase in solid concentration due to drag in ionic movement. The second factor is the electrical field strength. Electrical field strength is the quantitative expression of intensity of an electric field at a particular location. Increased field strength results in increased movement of fluid through capillaries, which is directly proportional to electrical conductivity. Greater the electrical field intensity, higher is the electrical conductivity and faster is the rate of heating. It has been reported that higher field strength of 104 to 105 volt per centimeter resulted in effective inactivation of microbes. The third factor is the particle size. The rate of heating in ohmic heating is affected by the particle size. Heating time required to achieve same temperature rise increased with particle size. The electrical conductivity decreases with increase in particle size. This in turn the rate of heating decreases with increase in particle size. Foods having high density and high specific heat values are conductive to slower heating. Fluid viscosity also influences rate of ohmic heating. Higher viscosity fluids tend to result in faster ohmic heating than lower viscosity fluids. The next factor is the particle concentration. Particle concentration is another crucial factor in determining the heating rate of the two phases. Study shows that electrical conductivity decreases as the pulp concentration increases. Strawberry pulp with 14.5 degree Brix has greater electrical conductivity than pulp with 26.5 degree Brix. Also, longer heating time is required for the product with higher concentration. An improved rate of heating was reported in fruit juices with the decrease in concentration of the juices from 60 to 20%. Ionic concentration is also another factor which influences the ohmic heating process. Structural changes like breakdown of cell wall protopectin, expulsion of non-conductive gas bubbles, softening and lowering of viscosity increases the ionic mobility in the product. So, increased ionic mobility in turn increases the electrical conductivity of the product. It is found that more acidic fruit pulp heated at a faster rate than non-acidic products. With regard to the applications of ohmic heating, a large number of applications exist for ohmic heating. The potential applications of ohmic heating includes blanching, cooking, evaporation, dehydration, sterilization and pasteurization, fermentation, extraction, baking and thawing. The most common problems encountered in thawing of frozen food products are longer thawing time, excessive weight loss, decrease in the nutritional value of food, which depending on the increase in the ratio of leak water and unwanted microbial activity during thawing. Hence, ohmic heating can be innovatively used to thaw frozen foods placed between two electrodes and applying an alternating current to it. The frozen foods can be thawed rapidly at temperature ranges -3 to 3 degrees centigrade. The major advantage in ohmic thawing is negligible generation of wastewater due to volumetric heating. Thawing process is uniform and the process can be easily controlled. A study shows that ohmically thawed frozen meat sample showed reduced drip loss, improved water holding capacity and shorter thawing time than conventional thawing. And the next is blanching. Blanching is one of the important steps involved in fruits and vegetable processing in order to control enzymatic activity, reduce microbial population, improve rehydration, remove air pockets from tissues, etc. Conventionally, blanching is carried out in hot water bath, which resulted in loss of soluble solids. Conventional and ohmic heating of P. puri samples was investigated by researchers. During the study, the samples were exposed to 4 different voltage gradients in the range of 20 to 50 volt per centimeter. Also, the samples were heated from 30 to 100 degrees Celsius and held at 100 degrees Celsius to achieve suitable blanching. Inactivation of peroxidase enzyme was observed from 30 to 50 volt per centimeter voltage level at less time than the conventional water blanching. Also inferred that the ohmic blanching at 50 volt per centimeter gave shortest critical inactivation time of just 54 seconds. The color of the raw product was also well retained after ohmic heating, while after water blanching it changed significantly. The various advantages of ohmic heating are: volumetric heating avoids large temperature gradient within the foods and rapid achievement of high temperatures. Higher temperature in particulates than liquid can be achieved, which is impossible in conventional method of heating. Reduced risk of fouling as there is no hot surface for heat transfer. Minimal mechanical damage to product. Better nutrient retention. High energy efficiency and low capital cost. Ease of process control with instant switch on and shut down. Reduced maintenance as there is no moving parts, and environmental friendly system. The ohmic heating process has its own limitations or challenges apart from its advantages. They are: difficulty in temperature monitoring techniques for locating cold or hot spots in continuous throughput systems. Differences in electrical conductivity between the liquid and solid phases and their dynamic response to temperature changes, which cause an irregular heating pattern, and complexity and difficulty in predicting or modeling heating characteristics of the particulates in the carrier medium. Lack of data concerning the critical factors such as resident time distribution, particulate orientation, ratio of electrical conductivity and loading rates. Let me summarize the points so far discussed. During ohmic heating, heat is generated within the food by the passage of an electric current. Ohmic heating is also referred as electrical resistance heating or Joule heating. Both liquids and particulate foods can be successfully treated in the short time with maximum product quality. Ohmic heating produces a sterile product. Application of electric current leads to permeabilization of cell membrane. Electroporation is responsible for inactivation of microorganisms and spores. Ohmic heating thus can be utilized in a wide range of food processing applications. Thank you for your patience hearing. Thank you.